Today we will be modeling the catalytic cycle of the enzyme hydrolase. An enzyme is a substance produced by a living organism that acts as a catalyst to bring out and induce a chemical reaction. Basically, this means that the enzyme induces the speed of the chemical reaction.
Hydrolase is found in intestinal tract and in saliva. Hydrolase is a hydrolytic enzyme that catalyzes the hydrolysis of a chemical bond and acts upon substrates. The product it creates is hydrolysis when H2O is added, meanwhile it converts.
Coenzymes are a type of cofactor and they are bound to enzyme active sites to aid with their proper functioning. Coenzymes which are directly involved and altered in the course of chemical reactions are considered to be a type of secondary substrate. There are two types of inhibitors when dealing with the catalytic cycle of an enzyme. Competitive inhibition is when the inhibitor and the substrate compete for placement in the active site of the enzyme. These inhibitors are said to have the same shape of that of substrate and prevent the enzyme and substrate complex from forming. This is done when the inhibitor enters the active site, thus preventing the substrate from entering. Therefore, the rate of reaction is decreased due to less substrate molecules binding to the active site. This type of inhibition can be overcome by increasing the substrate concentration. In the form of non-competitive inhibition, the inhibitor does not compete with the substrate for place in the active site, but however does reduce enzyme activity by binding to another site on the enzyme known as the allosteric site. Unlike the competitive inhibition, non-competitive inhibition prevents the formation of products from substrates. Another thing to know about these inhibitors is that they are unaffected by substrate concentration, thus most of them are permanent.
An allosteric regulation is the protein's function at one site and is affected by binding of a regulatory molecule to separate a site. Cooperativity is when one substrate triggers the shape change in other active sites and increases catalytic activity. Feedback inhibition is the end product of metabolic pathway that shuts down the pathway by binding the allosteric site of an enzyme and prevents wasting chemical resources. Allosteric inhibition is the binding of one ligand and decreases the affinity of a substrate at other active sites.